I'm Erica Berg, Director and Senior Editor for Custom Publishing at Science, and I'll be the moderator for this discussion. Autoimmune diseases are on the rise, and an estimated 1 out of 10 people may be affected. Effective treatments are needed for this vast and growing health problem. B cells have emerged as major players in the biology of autoimmune disease, and B cell depletion therapies are enjoying clinical success in the treatment of autoimmune diseases such as lupus and systemic sclerosis. Today, we'll be discussing why B cells are critical targets and how their depletion offers hope for better patient outcomes. Before we get started, I wanted to give a few instructions about the viewing console. Just to the right of the video window is the Presenters tab, where you can find photographs and biographies of today's speakers. You can log into your social accounts during the webinar to post updates about the event. On X, you can add the hashtag #ScienceWebinar. After the speaker presentations, we will have a short Q&A session, during which we will answer some of the questions submitted by our live online viewers. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab, also on the right, typing the question into the message box, and then clicking Submit. Finally, a thank you to Novartis for sponsoring today's webinar. I'd now like to take the opportunity to welcome our fantastic panel today. I'll give each of them a chance to say hello and introduce themselves. First, I'm pleased to welcome Georg Schett. Hi, Georg.
Yeah, you pronounced my name very well. I'm glad to hear that you took a lot of effort. It's a great pleasure to be part of this webinar. My name is Georg Schett. I am working in Germany at the University Clinic of Erlangen-Nuremberg. I'm interested in autoimmune disease and new ways to treat autoimmune disease, and B-cell depletion is an important tool to treat autoimmune disease. I'm looking forward to have an interesting webinar together with Thomas Dörner.
Thank you, Georg. Next, I'd like to welcome Thomas Dorner. Hi, Thomas.
Yeah, hello. Also from me, it's a great pleasure to be here together with Georg Schett as faculty. I'm interested in B cells since my postdoctoral stay at UT Southwestern in Dallas, and I'm working at the Charité and the German Research Center for Rheumatology, DRFZ, here in Berlin. And I'm trying to understand better the role of B cells and how we can selectively or at least semi-selectively target and use them for better outcome for our patients, in particular patients in need with very substantial systemic autoimmune diseases.
Great, thank you so much, Thomas. And you can begin your presentation.
Yeah. So in my talk, I would like to focus your attention on the role of, in particular, memory B cells and plasma cells. And although I will go in a little bit global overview, I will take the opportunity to use systemic lupus as a model for a systemic autoimmune disease where we are seeing an emerging and additional role for those two subtypes of B cell differentiation, memory B and plasma cells. Next, please. These are my disclaimers. Next, please. After a short introduction of B-cell depletion in several autoimmune diseases, I will go into the complexity of B-cell differentiation, have also subsequently a look into certain disturbances of B lineage cells in systemic lupus, in particular the hyperresponsive BCR signaling and the potential pathogenic positive feed-forward loop under the condition of lupus, and somehow trying to pave the way, in particular also to understand what Georg will share with you in order to come then to the current and emerging B-cell depletion therapies. Next, please. When we look into the different diseases where in particular CD20 treatment as one of the initial drivers of better understanding B-cell depletion, its effect, and also understanding on a reverse translational way their role in the pathogenesis, we have seen that in certain diseases, for example, that have been considered T-cell driven, like relapsing, remitting multiple sclerosis, some of the other neuroimmunological diseases, we have seen approvals in certain subsets of multiple sclerosis, but also approval for patients in rheumatoid arthritis who fail TNF inhibitors, that the treatment with B-cell depletion is really very effective. And on the other hand, somehow surprising, looked very safe as compared what would have been expected 20 years ago. We have seen some struggles with systemic lupus because maybe this has something to do with trial design, but also understanding better the role of one or the other B-cell differentiation stages. And I think this was a kind of initial possibility to look into the role of autoimmune B lineage clones. And the pioneers at UCL in the United Kingdom, like Joe Edwards, they wanted to go out in severe rheumatoid arthritis patients in order to eradicate B-cell clones, the clones that are producing rheumatoid factors. Now we know that this is possibly not happening because some of the B cells are really, or the underlying plasma cells are CD20 negative. But we learned a lot over the years also to balance what could be achieved in terms of the efficacy in one or the other autoimmune disease, but also what does it mean in terms of the immune protection, how we impair this. And in particular, the use of rituximab as well as MMF have told us under the condition of COVID-19 that this was really a very important risk factor. But we have learned a lot also from our patients that not all of the different autoimmune diseases responded in the same way to B-cell depletion therapy with rituximab. And we have learned step by step there are different segments of autoimmune conditions. And I think we are just beginning with the new emerging concepts to further segregate and develop a better understanding of the individual autoimmune diseases. Next slide, please. In the next few slides, I wanted to introduce, and you may forgive me if I'm recapitulating textbook knowledge, but I think it's very much important to understand the complexity of B cells. We are having, for example, as you see here on the upper part, a B1 lineage cell, where those cells and the hematopoietic stem cells are sitting in the fetal liver, partially also in the bone marrow, undergoing central and peripheral tolerance, and can then differentiate quite rapidly into natural antibody-producing plasma cells that are usually short-lived. The other, I think the main B lineage, and I think this where we know a lot, in particular in mice and humans, we are having the hematopoietic stem cell in the bone marrow, and those are differentiating via the transnational type 1, type 2 cell, either as marginal zone cells in the spleen, into short-lived plasma cells, also providing mostly IgM, natural antibodies, or, and I think this is the most important differentiation pathway, after encountering antigen and receiving proper T cell help, we see during germinal center reaction the induction of long-lived CD19 negative plasma cells or CD19 low expressing plasma cells that are rather short-lived, but we see also plasma cells and the induction of memory cells. And the last differentiation, I think, is very important also to understand what is happening in most of the autoimmune diseases, where we are seeing long-lived plasma cells with long-standing autoantibody titers. Next slide, please. When we look into what is happening in terms of the genetic re-engineering, I think B cells are those cell subtypes among all of the immune cells that undergo substantial changes on the DNA level. We see during recombination in the bone marrow that DNA rearrangements are forming the variable gene regions, the VDJ regions for the light as well as the heavy chain, prone for error, and a lot of those cells are not coming really out of the bone marrow because they are incapable of binding antigen or having stop codons, etc. After encountering antigen and receiving proper T cell help, we see a next step. Please, the next slide, please. And in the next step, we see further, let's say, processes again on the DNA level with immunoglobulin switching, receptor editing, as well as class switching. Again, on the DNA level, prone for errors, but this is very important in order to serve the concept of affinity maturation. There have been a lot of questions how this is maybe abnormal or differential in terms of autoimmune diseases. Can you please click the next? But after a lot, we have learned a lot from the underlying mechanisms, in particular the involved proteins and enzymes, etc. The initial concepts that maybe in one or the other autoimmune disease these molecular processes are abnormal or differential has not been proven over time, with very few exceptions, and I'm happy to talk about this. But I think this initial concept, mainly with the early studies on single-cell PCR data, have already falsified. And therefore, what we are seeing is most of the time really the impact of extrinsic factors forming an abnormal B-cell repertoire in most of the autoimmune diseases, including the production of autoantibodies. Next slide, please. So the very important question is, where are all these molecular processes out after the bone marrow, after recombination are happening? I think there is the concept, mainly under the involvement of TFH cells and the instruction by IL-21, we see conventional germinal center reactions where we see the induction of long-lived plasma cells apparently producing long-lived autoantibodies. Pretty much important, we see here a light zone and a dark zone, and in particular in this lymphoid follicles or the germinal centers, we have FDCs, and the FDCs are very important in order for negative selection and controlling what is coming out here. And apparently an early induction of autoimmunity where we see the long-lived plasma cells induced, these FDCs are allowing the emergence of autoreactive clones. What you see in the upper part, and this is in extrafollicular sites and in particular in the tissue, we see mainly under the involvement of tissue-resident peripheral helper cells. And a nice study has been published in this summer by our colleagues at Yale, that they have identified certain TPH cells thriving in the tissue, but also at extrafollicular sites, the induction of certain autoreactive B cells as well as autoimmune plasma cells. Next slide, please. A very important observation is that this is not happening at extrafollicular sites. We are seeing also these aggregates in the involved autoimmune tissues. Here's an example from a lupus nephritis kidney. You see aggregates of T cells surrounded by B cells. Those B cells, so those T cells are activated, they express ICOS and apparently able to induce the local B cells to differentiate into plasma cells. And those plasma cells are producing IgA, IgM, and IgG. And what is again missing here are the FDCs, where there is a possibility to control the emergence of autoreactive B cells or plasma cells. Next slide, please. So what are the disturbances of B-cell differentiation in lupus? And I will also look a little bit in the potential comparison into the characteristics of a secondary immune response, so the reactivation of already preformed memory B cells able to further differentiate into plasma cells. Next slide, please. When we look into what is happening in the peripheral blood of lupus patients, you might appreciate on the right-hand side on this dot plot, there is an expansion of plasmablasts. And we discovered this a couple of years ago. The occurrence of plasmablasts are very nicely correlating with the disease activity in lupus, but also in other diseases. They are even better than anti-DNA antibodies. But we saw also here in the red circle an expansion of so-called double negative atypical memory B cells. If you could click for the next slide. Over the years, Iñaki Sanz and other colleagues did a lot, also including our lab, in the further investigation of these atypical double negative B cells that are usually IgD negative and CD27 negative. You can look into a lot of markers, and I think most of those cells that are included here are memory cells, but we also found with the use of CD19 and CXCR5 that some of them are already pre-plasmablast. Some of them are still naive B cells. Happy to go into the details, but you may appreciate the abnormality in the peripheral blood already. Maybe we can click on the next slide. But we had then the possibility to look into the situation of COVID-19 patients who were infected, as well as patients undergoing mRNA vaccination. And what we really could find under the condition of vaccination, and we did also years ago tetanus, diphtheria, and others, usually with the proteins, you find what is the textbook telling us, the conventional differentiation of activated naive B cells becoming germinal center cells. And then you have a good output of conventional memory cells as well as plasma cells. And those plasma cells apparently who made it through the germinal center reactions, most of them are CD19 negative, considered long-lived. Some of them are also CD19 low expressing. Could you click please on the next? What really became apparent, maybe also the next one, when we compared our insights into patients with systemic lupus, as well as patients who contracted the infection or had the mRNA vaccination, we saw an expansion, and not only we, others too, of so-called double negative atypical memory cells, but also most of them are only inducing, or this under the instruction of other cytokines like interferon alpha, type 2 interferon, but also IL-6 and other cytokines. Most of the outcome in terms of the plasma cells are so-called CD19 low expressing plasma cells, what we consider a subsegment of plasma cells that is not long-lived or the longest-lived plasma cells subsegment. It's rather shorter-lived or short-lived. What we believe that we see some similarities with this situation with an mRNA vaccination as well as a virus infection with an mRNA virus compared to a conventional tetanus or protein induction, that we see the very important role of different T follicular or T peripheral helper cells, that the inducing antigen or autoantigen plays an important role, as well as the immune activation site, because here in the upper part, most of the immune activation is happening at the extrafollicular or in situ in the affected tissues. Next slide, please. We gained a lot of interest in the different segments of plasma cells a couple of years ago, and we found initially that CD19 negative bone marrow plasma cells are really aggregated or concentrated in the bone marrow, what you see on the left diagram. It's not accumulating in the spleen, other lymphoid tissues, and not substantially found in the peripheral blood. In some patients with rheumatoid arthritis, together with colleagues in Gothenburg, we had the possibility to look in patients with RA undergoing rituximab treatment. And what you might see here in the middle diagram, CD19 negative, considered long-lived plasma cells, did not change under treatment, but the CD19 low expressing plasma cells really declined here on the significance level of 0.05, lower than 0.05. And we believe that these shorter-lived plasma cells expressing a little bit of CD19 are really depending on the replenishment from the periphery. When we had the possibility to look into the bone marrow of some lupus patients, but also in the CSF of multiple sclerosis patients, we did not see a black and white picture, but there was a higher aggregation of DNA-producing plasma cells in the CD19 low-expressing plasma cell segment. Next slide, please. This gained also further interest, and we looked then into, in the meantime, we looked into several hundred bone marrows of healthy individuals not suffering from autoimmune diseases, but undergoing hip or knee replacement. And what you might here see is the separation in the dot plot between CD19 negative and CD19 low expressing plasma cells. And when you look how, for example, tetanus versus RBD, so SARS-CoV-2 related immune responses, how the different plasma cells are residing, you might appreciate that the tetanus ones are really located in the CD19 negative plasma cell compartment, and the RBD specific ones are rather aggregated in the CD19 low expressing, so short-lived plasma cells segment. Next slide, please. But we went further together with Farzin Mashregi and his group. We looked into an unbiased way on a single-cell RNA level, and you see here altogether 10 different clusters. In the mouse, Andrea Lino in our group found 13 different clusters. And the cluster number 1 here, this is the CD in blue, the blue dots. This is the population of CD19 negative bone marrow plasma cells. And most of the other ones here are CD19 low expressing plasma cells, the other 9 plasma cell segments. Maybe you can click one further. But when we looked into the different immunoglobulin classes that are residing in the different clusters, together with certain signatures of cytokines, we found in particular for the cluster number 1, a substantial signature of IL-21, type 1 interferon, and TGF-beta, and all in the other ones, certain different cytokines where we believe what we see here is at least for the moment, and we are working further on this to understand this, that we see what is happening or what was happening before in the initial immune responses. And this is a kind of integral of the prior immune responses, what we are seeing here in the bone marrow in terms of the different immunoglobulin classes and the correlating signatures of certain cytokines. In the moment, we are studying this also in patients with systemic autoimmune diseases, and maybe in one or the next of these seminars or whatever, we would be very happy to share the data with you. Next slide, please. The question is, when you look into the textbook, please click further. You find in the copy and paste, maybe you can click one further, that B cells in autoimmune models, in particular lupus, are hyperreactive. But we challenged a little bit the textbook because we found, and we struggled a lot, whether our data are correct or not. We found in terms of BCR activation and other activation statuses that those cells are hyperresponsive. We call it also anergic post-activated. How do we come to this? Maybe you can click one slide further. Before we go into this, you may appreciate that the B cells, and I think that is maybe one of the central roles that they can play in the condition of autoimmune diseases. It's one of the few cells where signaling pathways of the innate and the adaptive immunity are crossing and might be interregulated. Maybe next slide. When we compared the activation of BCR signaling downstream of the BCR signaling molecules like the phosphorylation of spleen tyrosine kinase SYK or Bruton's tyrosine kinase BTK, and as a reference, AKT phosphorylation up in BCR activation, we found a substantial lower amplitude. Here, all the colored lines are what you're seeing for Sjögren's, rheumatoid arthritis, and lupus. And maybe you can click one further. To make a long story short, you may appreciate here the dashed line. This is the activation of the B-cell receptor signaling pathways here for SYK, for memory B cells. From normals, the black line, you find this for naive B cells. And if you look for naive and conventional memory cells from lupus patients, you see the red lines. The difference between the memory B cell and the naive B cell activation is gone, and the amplitudes are much lower. We did this for dozens of patients, and I can only tell you this is highly, that is highly reproducible. We struggled a lot, and maybe you can click further to understand this. How can we understand how this can be overcome by one or the other stimulation? Can you please click further? And we found initially, when we looked into potential differences, that there is a higher transcriptional activity of receptor type protein tyrosine phosphatases that are apparently counteracting the phosphorylation of the BCR signaling pathway, most pronounced in lupus. When maybe you can click further. When we did then stimulate, and we did a lot of different stimulation pathways, but in particular, when we stimulated CD40, the main, let's say, change what we found is a downregulation of most of the receptor tyrosine phosphatases. If you please click further, but in addition to the transcriptional changes, we found also then up in CD40 stimulation that the response of the BCR signaling for BTK, AKT, and PCL gamma almost normalized, so that we can assume that this is one pathway where these B cells are still, apparently by bystander T cell activation, can still respond and, let's say, take part in the immune activation. Next slide, please. The other interesting, and you might also already click further, the other interesting observation was in an experiment when we stimulated PBMCs from different patients with CD3, and overnight, after 16 hours, we separated B cells, T cells, and K cells, etc. And you may appreciate here on the upper right corner that after the 16 hours of simple T cell activation, we found an increase of the type 1 and type 2 interferon gamma expression in B cells only. And on the other side, a downregulation of the Fc gamma receptor pathway, where we think that there is an abnormality how T and B cells are interacting. And one of the important signatures as an outcome, a very simple experiment, but I think it's very instructive to see the abnormality that these B cells are responding with an enhancement of interferon gamma signatures. Next slide, please. How to bring these things together. And we believe that we have an anergic, when we compare this on the left-hand side with a normal situation where you have a normal BCR response after in BCR activation, we have an anergic BCR response. But this anergic BCR response can be overcome by, on the one hand, bystander T cell activation by a CD40, CD154 interaction, leading to an NF-kappa B activation. Alternatively, with TLR7, TLR8 ligands, that can either also signal through NF-kappa B or to an IRF7 signaling pathway. Maybe you can click further. And possibly the B cell receptor can passively take part, internalize certain RNP, whatever, but co-ligate with TLR activation. But what we are seeing is apparently a selection of most of the RNP antibodies via TLR7, TLR8. We might also be able to explain a female preference, for example, in lupus because of the incomplete X chromosome inactivation, in particular for TLR7, TLR8, and CD154, which is the ligand or the counter ligand for CD40. Both, all of them are encoded on the X chromosome. And we also think that this might be an important explanation of the interferon gamma signal or the interferon type 1 interferon signature, mainly related to TLR7, TLR8 activation, in particular in lupus. Next slide, please. When we bring this together and put the memory cell in the center and the abnormality, we can somehow also come up with a proposal, with a model that these abnormal memory B cell activation, where primed memory B cells are able to either receive bystander T cell help via CD40 or able to, let's say, allow the expansion of TLR7, TLR8 ligand-dependent activation, maintaining or inducing autoreactive anti-RNP antibodies that are able, via the forming of immune complexes, to activate PDCs. And these PDCs are really firing type 1 interferon that is also able to activate the myeloid or monocyte macrophages, and that are, on the other hand, induced to produce type 1 interferon, but also BAFF and APRIL, very important to maintain memory B cell as well as plasma cell survival. Please click one further. Coming now to the therapeutic considerations, we have seen that with, and this is from a recent submission from Tobias Jund, and I'm very happy that he shared this with me. We are really, and you may see this, the coverage of CD20, we are now going more into earlier B cell targeting as well as more into the plasma cell targeting with one or the other molecule like BCMA, expanding CD20 further by using CD19 as a target molecule, but also using CD38. A very, let's say, or less selective treatment strategy where I think this is a good model is autologous stem cell therapy, where we have collected a lot of experiences over the last years. Please click one further. Before we go into this, I simply wanted to raise here the point, on the one hand, on the left-hand side, with the antibodies type 1 CD20 antibodies like rituximab, ocrelizumab, ofatumumab. On the one spectrum, we have learned over the last 10, 15 years a lot in terms of their efficacy as well as their safety. And on the other hand of the spectrum, there is autologous stem cell therapy, where we, after conditioning, eradicate or deplete completely the immune system and allow immune reconstitution. But in between, with glycoengineered antibodies, as well as with T cell engagers now, as well as with CAR T cells with very different targets, we think that we are now in a possibility in the spectrum between stem cell therapy and CD20 to match one or the other autoimmune disease or optimize efficacy and safety. We might also be able to tune one or the other treatment strategy for one or the other autoimmune disease, and we will learn a lot in terms of reverse translation, how the different mechanism of autoimmunity in one or the other autoimmune disease is functioning. Next slide, please. In my last slides, I would like to focus your attention again on the memory B cells, because I tried to convince you that they are very important. And a couple of years, Mark Schlomczyk came up with a mouse model based on a lupus background, but these mice were not able to secrete antibodies. So they could also not secrete autoantibodies. And I think they brought forward a very interesting concept that let's say, the role of B cells per se, without their function as precursors of antibody-producing plasma cells, are really very important. Maybe you can click further. What we also learned from stem cell therapy, but also type 1, type 2 CD20, we still learn this from ianalumab, an anti-BAFF receptor antibody, and I think the data so far are also consistent for the available CD19 and BCMA CAR T cells. We are still waiting for the T cell engagers, how they perform in terms of the recurrence of memory B cells, because I think the depletion of memory B cells in the tissue and the, let's say, disruption of their detrimental function is very important to best control that or preventing a recurrence of the disease. Let's go in a few examples. Before we go into this, I wanted to raise your attention on this interesting 5-year survival or progression-free survival data in patients with different autoimmune diseases after autologous stem cell therapy. You see almost superimposed curves for lupus, systemic sclerosis, multiple sclerosis, and GIA, having about 70% 5-year progression-free survival. The only curve that really is different is rheumatoid arthritis. It has a much poorer survival, and I think it already teaches us something about the differences of rheumatoid arthritis when we are talking about treating the immune system. Maybe we need to better understand the stromal cells and their function, how they are allowing the immune system to, let's say, maintain their abnormal activity. Next slide, please. We have seen in a couple of patients one very instructive case with a relapse after stem cell therapy, and the relapse happened about 18 months after the initial treatment. Please, next slide. Most of the patients are really in remission, and that's fine. But this patient, prior to the clinical reoccurrence of systemic lupus, had an increase of T and B cells, in particular T and memory B cells. And this was a very important and I think instructive finding. The earliest abnormality that was seen prior to the clinical recurrence was really the recurrence of memory cells, not antibodies, not the recurrence of T or B cells when they are naive. Next slide, please. But when we are talking about stem cell therapy and also the other very broad depleting therapy with Campath-1, the CD52 alemtuzumab treatment, we see a substantial impairment of the protective immune globulin titers because we are wiping out the bone marrow, all the plasma cells are gone. Next slide, please. And the other issue, and I think we are now in a beautiful situation to learn more when we are depleting B and T cells together. The early cells that are coming back are the T cells. And when they are around without B cells, we are seeing the challenges in stem cell therapy as well as in alemtuzumab treated patients of secondary autoimmunity. ITP, autoimmune hemolytic anemia, autoimmune thyroiditis, and very important and sometimes really life-threatening acquired hemophilia A. This apparently is not happening if you selectively deplete B cells or plasma cells only. Next slide, please. So simply share you and convince you further that that is important to look into memory cells. You may appreciate this is from the phase 2 nobility study using a second generation CD20 antibody obinutuzumab. So you see, after the 2 cycles of treatment, on the left-hand side, naive B cells are coming back almost normal after treatment. They are similar as in the patient who received a placebo. The memory cells are staying really down, and most of the patients are really responding, and the response is here really also related to the depletion of apparently tissue-resident cells, and including memory cells, and the recurrence of plasmablasts or plasma cells did not matter here in this situation. Next slide, please. Let me conclude. I do hope that I could convince you that B lineage cells, and I think Georg will follow on this, continue to be very attractive treatment targets beyond CD20 and across different autoimmune diseases. I think memory B cells are pretty much important, at least in lupus, because they are really at the crossroads of innate and adaptive immune signaling. They can form together with autoreactive plasma cells a positive feed-forward loop via anergic the B cell receptor signaling, but still able to respond to TLR7 and 8 signaling and CD40 activation. Sustained clinical response is really inversely related to memory B cell repletion or reoccurrence. And I think more selective B lineage depletion using advanced effector mechanisms, for example, with FC engineered antibodies, CAR T cell or T cell engagers, as well as selective targets, CD19, BCMA, provide opportunities for deeper and broader depletion. And I think we will also be able to advance our understanding for certain autoimmune diseases on the one hand with selective efficacy, but also in terms of safety. And I think in reverse translation, we will learn a lot from the normal immune system, in particular on the B lineage side. With that, maybe next slide. I need really to acknowledge for a long time a lot of collaborators, including my own group and national, international collaborators and local here in Berlin at the DFZ, at the MDC, and the funders, in particular, the German Research Foundation, DFG. Thank you so much.
Thank you so much, Thomas. And Georg, you'll be up next. Lots of questions have been coming in online. Keep them coming. Lots of interest. So thank you so much, Thomas, and off you go, Georg.
Yeah, thank you. Thomas has very nicely alluded to the importance of B cell in autoimmune disease, and has also gave rise to new ideas and concept how to deplete B cell from patients with autoimmune disease. And I want to follow up on this and discuss the current dilemma what we have in autoimmune disease, and what are potential ways to improve overall therapy. Next slide, please. So it is going back to Paul Ehrlich coining the term horror autotoxicus, indicating that if the immune system recognizes its their own body, the body is in a serious trouble, and that's actually what we observe in autoimmune diseases. Usually they are severe, they are lifelong, and overall destroy the tissue architecture of the affected organs. Now, Paul Ehrlich didn't know about all this antibody specificity, but basically autoantibody generation and deposition or target cell engagement leads to a destruction of physiological process or interference with physiological process, which then lead to inflammation and organ dysfunction. Next slide. So this creates what we think is so-called autoimmune enhancer loop. And this autoimmune enhancer loop is making actually an inflammation chronic and sustained, in contrast to an inflammatory response, which can be seen as a physiological response. So the sustained inflammation is actually the key process which drives pathology in many of the autoimmune diseases Thomas has mentioned, like lupus, for instance, and rheumatoid arthritis. And this autoimmune enhancer loop is crucially dependent on a B cell activation against self with the deposition of antibodies in the tissues through the generation of plasmablasts and plasma cells as the key antibody producing cells. Next slide, please. So this slide shows the important observation that autoantibody generation precedes the inflammatory phase of many autoimmune diseases. Here, as an example, systemic lupus, and that's mimicked by several other autoimmune diseases where autoantibodies and autoantibody maturation, that means the increase in the levels of autoantibodies, the epitope spreading, that means that you get a broader autoimmunity, and the generation of new autoantibody entities is a typical feature in the years before the patient actually gets sick, consults the doctor, and requires treatment. Next slide. So this is a very busy slide, but it just indicates that this concept of B-cell targeting in human medicine is not new. It's actually old and has led to multiple trials in very different autoimmune diseases targeting and depleting B-cell. And here is a list of typical studies in autoimmune disease based on B-cell depletion. The most common target is CD20, a B-cell specific antigen, which is targeted by rituximab and other more modern antibodies, as Thomas has alluded, for instance, ocrelizumab, obinutuzumab, ofatumumab. So there is actually a very busy space to target CD20. And apart from that, there are also other antigens which are interesting in B-cell depleting therapy, like CD19. You can see that there is a broad spectrum of diseases which has been tested for B-cell depletion. Some of them very successful, like rheumatoid arthritis, like epidermolysis pemphigus, for instance, or ANCA-associated vasculitis. Some of them showing much more resistance to B-cell depletion, like lupus, for instance, or Sjögren's syndrome, suggesting that there must be there's a certain complexity in B-cell targeting which has not been fully understood to date. Next slide. And this complexity is indicated by the fact that, there's just one example, that a typical paradigm autoimmune disease based on B-cells like systemic lupus has not shown dramatic efficacy in randomized controlled trials by targeting B-cells with rituximab. So this is a negative study here, the Explorer study, showing no difference between rituximab and placebo in lupus erythematosus, which has been very puzzling, I think, for the research community, because everybody thinks, and based on the strong autoantibody background of lupus erythematosus, it has been very puzzling that such approaches to deplete B-cells have not been more successful, more robust, and more sustained in lupus. Next one. And the reason for that is most likely the fact that you can't easily deplete tissue B-cells in the body by an antibody like rituximab. But that has been shown multiple times, for instance, in bone marrow biopsies from patients with rheumatoid arthritis, in tonsil biopsies from patients with lupus, and in synovial biopsies with patients with rheumatoid arthritis. It has also been shown, like this slide indicates, in transplant medicine, where rituximab has been used to avoid humoral transplant rejection. And here this slide shows in the gray bar the persistence of CD19 positive B cells after rituximab exposure in abdominal lymph nodes, indicating a substantial resistance of these cells to get killed by a drug like rituximab. Next slide. So what we do actually with rituximab, which is by far the most widely used B-cell depleting antibody, is a shallow depletion of B-cells, mostly in the circulation, where the depletion is very good because you have effector cells allowing killing of cells by rituximab, but actually not going to a so-called zero B-cell state, which would be actually at the very bottom of this grid, but rather depleting only a small fraction of the B-cell, which means that when the drug is gone, the B-cell actually will come back, and also autoimmune B cells will come back, because autoimmune B cells, as many other B cells, survive the attack of this so-called B cell depleting antibody. Next one. And therefore, actually, new ways have been designed to deeply deplete B cell. That's one way, is actually to create higher affinity antibodies with better B cell depleting efficacy, as Thomas has mentioned. But also there are other ways to increase the effector function of the system. One is the so-called T cell engagers. They are allowing the bind. These are compounds binding to B cells, but also at the same time engaging T cells, creating a deadly rendezvous between B and T cells, and allowing a good B cell depletion. And then the other one is CAR T cell therapy, whereby actually the T cell is used as a living drug, expressing the CAR, binding the B cells, and killing the B cells by combining actually antigen recognition function and effector function in the same drug, in a therapeutic cell. And I will share with you some new findings in the field of T cell engagers, which are very recent, but also the findings of CAR T cell therapy in autoimmune disease. These are limited results yet, but I think these results are stimulating and suggest that this might be an interesting approach for the future therapy of autoimmune diseases. Next slide, please. So T cell engagers have been described in 2000. So it's not a new drug. In fact, it's 25 years or 24 years old. And a typical way to create a T cell engager is to take actually the antigen recognition part of an immunoglobulin, recognizing CD19, for instance, or another B cell antigen, and CD3, which is expressed on the T cell. So by combining these approaches, the molecule can actually liaise B cells with T cells. And the first example has been blinatumomab, which is a small molecule which has a short half-life and engages CD19 and CD3. And by this engagement, the T cell is used to kill the CD19 expressing B cell. Next slide. So as said, this story goes back 24 years, and the initial publication, it was very nicely shown that the T cell, that indeed such molecules drive T cell activation. So you can see the positive control on the top, which is IL-2. There's very robust T cell proliferation. And then you can see the fourth graph shows actually that you require B cells to allow T cell proliferation. So that's the black bar. So when B cells are present, the T cells are engaged and proliferate in the presence of this engager. With the control molecule, they don't do that. So you show that if you put B cells and T cell together, this molecule leads to a T cell activation. And at the same time, in the lower graph, you can concentrate on the black bars. You can see in the different lines of B cells, there is a robust killing of the B cells when they are exposed to a T cell engager. Next slide. So now this concept has actually been very interesting. And when you advance the slide, please, there are several molecules which have been approved for the therapy of lymphoma and leukemia. Next one, please. Next one, please. Next one, please. So you can see here 3 examples of molecules which are targeted by T cell engager. One is CD19, as mentioned, but there are also T cell engagers targeting CD20. As you know, that's a typical molecule on B cells. It's the target of rituximab. But there are also BCMA engagers developed and approved for the treatment of hematopoietic malignancies, particularly multiple myeloma, because this BCMA antigen is also found on plasma cells. Next one. So we started recently to explore the possibility of T cell engagers in the treatment of autoimmune disease and selected treatment-resistant rheumatoid arthritis patients. You can see here, this is a small group with 6 patients, extremely treatment-resistant rheumatoid arthritis patients with very active inflammation, and they were exposed to a very tiny amount of a T-cell engager, in this case, blinatumomab. So to say, these are doses in the microgram level, 1,000 times less than with any therapeutic antibodies currently approved for the treatment of rheumatoid arthritis. Next slide, please. And what we observed is an engagement function, which is very interesting. You can see here that with the first course of blinatumomab, which is an IV infusion, the C-reactive protein goes up. That means that that's an indirect sign for a kind of inflammation happening after the T-cell engager. And this is easier seen also in this kind of colored cytokine expression pattern, where you can see in the second column that there is much yellow, and that's actually the inflammation, the T-cell engagement happening, which means there is some cytokine release. But then when you look at the last column at the right, you can see a lot of blue and violet. So the cytokines expression goes down massively, especially as compared to the initial baseline situation. So you can see a time-limited engagement function, which activates the T cell and allows the killing of the target cells, namely the B cell, in rheumatoid arthritis patients. Next slide, please. When you look at the B cells, of course, B cells are depleted in the periphery. That pertains to naive B cells and memory B cells. The B cells come back over time, particularly more naive B cells than memory-based B cells, suggesting that there is a reconstitution of the B cell compartment from the bone marrow and some kind of reset of the pathologic B cell system upon engager therapy, which is, by the way, also seen with standard B cell depleting antibodies, but it shows that there is a clinical or a biological effect of the engager on the B cell system. Next one, please. What is also important that you can very nicely monitor the T cell engagement in vivo in humans. And here you can see CD4 cells and CD8 cells. You can see the first dose and the second dose. And you can very nicely see the engagement of CD8 and to a lesser extent CD4 cells. So they are consumed, they are actually attracted to the site of work, which is actually where the B cells sit. It's mostly in the secondary lymphatic organs where they do the killing function. Next slide. And the clinical efficacy of this approach was very good. We had a decrease of the swollen joint count and overall disease activity score in this very resistant rheumatoid arthritis patient. This effect was not sustained. It has to be then maintained by other anti-rheumatic drugs, but it shows a proof of principle that it's possible to use engagers in an autoimmune disease like rheumatoid arthritis. Next slide. The second approach for engagers is to target BCMA. As mentioned, that's a typical target on plasma cells, and we still think, and Thomas has done a lot of work on that, that some diseases might be very much anchored on the plasma cell, and destruction of plasma cells might be required in some autoimmune diseases. Next slide, please. And what has been recently shown, that's really very recent data, that this BCMA engagers seem to work also in autoimmunity. There is one publication from Berlin by Tobias Alexander showing very nicely that a BCMA engager, in this case teclistamab, is effective in resistant lupus erythematosus. And you can see basically very nicely upon the administration of this engager, the lupus disease activity went down, double-stranded DNA antibody went down, and also signs of plasma cell presence went down when this patient got the engager. Next slide, please. That seems to be important also in other autoimmune disease. And here is a case series also recently published by our group showing very severe patients with systemic sclerosis, severe cases of myositis, Sjögren's syndrome, and extremely refractory rheumatoid arthritis patients, who have been heavily pretreated with various drugs. that BCMA engagers might provide benefit to these patients. Next one. This is BCMA engagers also lead to a time-limited increase of C-reactive protein level and cytokines, as you see here, which is not surprising because also this compounds engage T cells. Next slide. And what is a bit surprising that actually BCMA targeting also depletes B cells in the periphery, suggesting that BCMA might not be uniquely expressed on the plasma cell lineage, but also on B cells, which explains the depletion of peripheral B cells in the circulation. But what you also see that it has impact on plasma cell compartment with a downregulation of the levels of the free light chains and immunoglobulins, which is owed to the fact that plasma cells are targeted. The T cell engagement function you can see here in the U-shaped form of the curve with the depletion or a reduction of peripherally circulating T cells, and also IgA levels, other parameters of adaptive immunity go down. Next one. What was reassuring to us that several disease activity parameters which are specific for these diseases, like the SDI and the DAS28 score, were actually going down after BCMA engagers. So suggesting that this is also an interesting tool to treat autoimmune disease. Next slide. Advance, please. Further advance. Next slide, please. Further advance. Okay, then how about CAR T cells? CAR T cells, as mentioned, provide a tool as a living cell to tackle B cells in a very deep way. Next slide, please. And this is usually done as an autologous CAR T cell approach. You can advance the slides now, please. Perfect. More. And again, so basically this, no, go back one, please. So this is the principle of autologous CAR T cells. You take actually leukocytes from the peripheral or central vein of the patient, you enrich the T cell, you basically transfect this T cell with a lentiviral vector encoding for the CAR. You actually expand these T cells in the ex vivo situation, and then give back a tiny amount of these autologous CAR T cells into the patient. And most of these CAR T cells used in autoimmunity target CD19, as you have heard before, which covers actually a large variety of the B cell lineage. Next one. And at the moment, I would say about 100 patients have been treated worldwide with CAR T cell therapy in autoimmune disease. In our own cohort that has been published comprises 15 patients. And these patients surprisingly had a very deep response and long-standing response without the necessity to treat them with glucocorticoids and immunosuppressive drugs. We treated lupus patients, myositis patients, and systemic sclerosis patients with CD19 CAR T cells and showing actually sustained responses. And these responses, next slide, please, were also very deep, as shown here, that in lupus you get actually a very strong effect on the disease activity and the seroconversion of the double-stranded DNA antibodies, leading to a dramatic improvement and rather fast improvement of symptoms in these patients. Next slide, please. That was also seen in very severe cases of dermatomyositis with lung involvement. Both actually the muscles and the lungs, and especially the interstitial lung disease, did respond to CD19 CAR T cell therapy. And in the next slide, you can see that there are also responses in severe systemic sclerosis patients, which is still a disease which is very hard to treat, and especially the lung and the progressive lung and heart involvement, which you see in the right, indicated by FAP PET, combined with a severe progression of the skin involvement, is a huge problem in this patient. And also here, there is a very sustained effect of CAR T cell therapy on these severe patients. Next one. We explain this sustained effect by a very thorough depletion of the B cells in the tissue. And we see here, when you remember the first graph with the antibodies, you can see here a hypothetical curve. What happens that the B cells indeed are depleted in the tissues, allowing actually to eradication of the autoimmune B cell clones and actually a recurrence of a naive, completely naive B cell system in these patients. Next slide, please. This concept has now been much more fostered by lymph node biopsies in patients with autoimmune disease undergoing CAR T cell therapy. And here you see one example of a staining for CD19 and CD20, where you can see a complete eradication of the B cell lineage in the lymph node, which is actually not found in rituximab treated patients with zero circulating B cells, which you see in the lower part of this figure where B cells are still present. So what we think that you get indeed a deep tissue depletion of the B cells, allowing this reset of autoimmunity and the eradication of the autoimmune B cell clones, and therefore a zero conversion of many antibodies, except probably antibodies coming from the plasma cell compartment. Next slide, please. And the overall safety of this approach seems to be surprisingly good. A cytokine release syndrome is usually at a low grade, cytokine release syndrome grade 1, which means fever indicated here without a lot of hemodynamic consequences. Next slide, please. And the fact that next slide, please. And the fact that you can actually stop glucocorticoid therapy and immunosuppression is very interesting, or potentially important for infection risk, because overall infection risk in the long run in these patients seems to be low because there is no concomitant treatment anymore, and also the B cells recover. This also is important for instance, pregnancies, because many of the drugs used in autoimmune disease are not compatible with pregnancy. It has to be stopped, and therefore the fact that there is a drug-free remission, sustained drug-free remission, might be important, especially for young patients with autoimmune diseases. Next slide, please. And I think what we have learned that since this approach seemed to work, there is a large expansion of CAR T cell studies recently in autoimmune diseases pertaining to lupus, but also pertaining to neuroinflammatory diseases like multiple sclerosis and myasthenia gravis, which also seem to respond to CAR T cell therapy, and where the therapy seems to be tolerable. Next one. In summary, what I wanted to share with you, there is an exciting development in B cell targeting strategies in autoimmune disease. I showed you the limitation of rituximab, not saying that rituximab is a poor drug. It's a good drug. It has been good. It changed actually the outcome of patients with autoimmune diseases. But now there are newer tools available, especially T cell engagers, allowing deeper depletion of B cells, but also CD19 CAR T cells, which lead to a long-standing drug-free remission in patients, which has been unprecedented in earlier times. Next slide, please, and final slide. At the end, I think what we see that our tools allowing also very resistant patients to enjoy remission. Some of these patients do not need to take any immunosuppressive drug regimens anymore. And I think we stand in the front of an exciting future where the cooperation between different disciplines in medicine can lead to a better life in such severely affected young patients. Thank you for attention.
Thank you so much, Georg and Thomas. I hope you can stick around. We're at time, but I wanted to ask a few questions if you're available. Is that okay? Okay, great. Starting off, I believe this one was for you, Thomas. Was the monitoring of peripheral memory B cells in blood sufficient, or did you require showing memory B cell depletion in tissue? Thank you.
Actually, for the already existing data, with the exception of Georg's study in from the lymph node biopsies, I think there were a few studies, but in very limited patient samples to demonstrate that there is residual memory cells in the tissue. Usually, the monitoring was done and is still done because of practical reasons in the peripheral blood. There might be possibilities with advanced technologies if you look into certain, let's say, molecules expressed by mostly tissue-resident cells, that you can see a kind of overflow of memory cells coming from the tissue. But this has not been 100% in the humans proven, for example, by FCLR4, which is it's a particular surrogate parameter developed by Professor Cooper a couple of years ago. But this has not so far been translated into clinical practice, and it's not done for monitoring in clinical studies.
Great, thank you. This could be for either. What is the difference between autoimmune diseases that readily respond to B cell depletion and those that do not?
Thomas, go ahead.
I think, but I tried to mention that we learned a lot of the responses in different autoimmune diseases, because with the availability of rituximab, there was a flood in different diseases in order to segregate response versus not response. Most of the responding entities are at least characterized by a typical or characteristic autoantibody or autoantibody profile, indicating abnormalities of the humoral immunity. We have seen, for example, in seronegative arthritis, very poor responses, but this is very superficial. What I really think that with the newly available or emerging therapies, going into different slices of B-cell plasma cell differentiation, we will learn a lot more how these diseases segregate in terms of the underlying mechanism, and which cells are really key drivers, need to be targeted by one or the other strategy. And this will also allow us to fine-tune the treatment.
Great, thank you. Gail, oh yeah, go ahead.
Probably another important piece for response is also that the level of damage, right, in individual patients. So if there is a lot of damage, it's harder actually to get good responses in the patient, and also the outcome parameters, right, are different, right? So in some diseases, outcome parameters are rather sensitive, but in other diseases, they are less sensitive. You probably need actually much deeper depletion of the B-cell system to see actually a response of a classical outcome parameter. So it's a complex story, right? It's based on the level of B-cell depletion or the way of B-cell depletion, but also available outcome parameters and damage. These are factors in deciding about response or not.
Yeah.
Great, thank you. All right, so looking ahead, what advancements in B-cell depletion therapies do you foresee in the near future, and how might these change the treatment landscape for autoimmune diseases?
Who starts, Thomas? Do you want?
Good question, but I think we also need to emphasize an important factor. On the one hand, I think we are looking all the time in risk-benefit ratios. Very important, we also need to bear in mind the safety. And if we, for a long time, eradicate plasma cells in the bone marrow, we are really impairing protective immunity. If this is delayed or long-standing, I think we are exposing the patients to a long-standing risk for opportunistic, non-opportunistic infections. And I think we have pushed the border now with the availability of eradicating plasma cells for a short time, for longer times. I think we also, on the other hand, need to look into safety considerations in addition to the efficacy. And sometimes it comes with surprises. I think 15 years ago, I never would have thought that multiple sclerosis, relapsing remitting, is accessible to B-cell treatment or CD20 treatment. And there, the damage has also already been substantially reduced in terms of the outcome. There are almost no gadolinium-enhancing lesions anymore after CD20 therapy. A huge surprise. But on the other hand, safety was very good and tolerable. And I think a very important aspect for me is also coming from the COVID-19 pandemic, where we have seen a lot of patients under rituximab who contracted very severe infections, and some of them we even lost because of this impairment. And maybe Georg.
No, I fully fully agree what you said, Thomas, and the risk-benefit consideration is always absolutely important. Having said that, I mean, I think gradually over the next 5 to 10 years, we will see a shift from the suppressive regimens to kind of resetting the diseases, right? So there's a key unmet need is that we have to usually treat the patient for a rather long time. And that's also the difficulty, right, for B-cell targeting. You blunt vaccination responses, you blunt immune response against infection. So you don't want to have this therapy necessarily over many years. You want to have actually a quick reset of the disease, and then a normal system builds up again, where you don't have to worry about so much about infections, pregnancies, and whatever. So I think this is where the future lies in, and we have to learn more how this is achievable. I think it is achievable. That we learned in the last couple of years, but we have to use that in a more stringent, more defined way, and I think that the next years will bring.